minus 5.9 micrograms per kilogram. Now, ricin toxin is a glycosylated heterodimer, and so this slide shows that we can express the RTA in E. coli and purify it, and I'm going to show you on the next slide that glycosylation isn't required for eliciting a protective immune response in animal models. And we use the A subunit as our target for vaccine development because it was known that the immunodominant epitopes, for the most part, reside on the A subunit. So you can make a synthetic gene, or you can use the native gene, express it in E. coli. You can purify the vaccine using ion exchange columns, very easy procedure to purify. And if you take that purified antigen and vaccinate animals, in this case mice vaccinated with IM injections at 0, 4, and 8 weeks, and then challenge either with a parenteral injection of ricin toxin. This is quite a bit of toxin, 10 mouse LD50s, or by an aerosol challenge using 5 to 10 LD50s of ricin toxin. The vaccine protects the animals completely. Controls die within, depending on the exposure route, by IP injection, the animals die between 6 and 20 hours. By aerosol, 3 days. But there's a problem with using the RTA as a vaccine candidate. And two of the reasons that it's a problem is, one, the N-glycosidase activity still remains in the RTA. And second, the RTA in itself is unstable, tends to aggregate upon itself, and over time precipitates out. And the reason for this is on the C-terminal end of the RTA subunit, you see a very large hydrophobic surface area. This region, as a toxin, is embedded into the B subunit, and these hydrophobic residues are hidden from solvent accent. So the ricin toxin itself is very soluble in aqueous environments, but you take it away from the B subunit, and then this doesn't like to interact with the solvent, aqueous solvent, so it tends to aggregate with itself and eventually precipitate out. So we had to deal with these two issues, the instability of the RTA, as well as the N-glycosidase activity still remaining in the RTA subunit. And so we used a molecular modeling to look at how to address first this hydrophobic effect that it's called. And we ended up removing C-terminal residues from the domain 3 of the ricin A subunit. And that domain 3 is from residues 211 to residues 267. So we essentially genetically remove the region, the blue region, which is residue 199 to 222, as well as the region in the yellow, which is residue 223 to 267. So essentially we remove residues 199 to 267, which really were the residues that were hydrophobic in nature, causing the instability of the protein. We also removed residues 33 to 44, which is this loop that just stuck out into the environment. We were concerned that this would be a target for proteases during manufacturing, so we just eliminated it. Still maintaining the neutralizing epitopes in domain 1, which is a residue between 90 and 110. And Nick Bantis has shown other neutralizing epitopes in the domain 2 of this molecule. Nick published a paper in Vaccine in 2010 showing these residues that we removed in domain 3 don't contain any neutralizing epitopes. They're all non-neutralizing epitopes. So we develop a scalable manufacturing process for this vaccine, which simply uses a cation exchange column followed by a negative anion exchange column, a second different type of cation exchange column, followed by a final diafiltration concentration step for bulking the PDS, or the BDS, bulk drug substance, product drug substance. We have now 24 different analytical and biological assays for characterizing this protein. I'm going to show you 2 of them. These 24 different assays are used in lot release of the vaccine. This is an SDS, reducing SDS page of the vaccine. This is 4 different lots of the vaccine that shows the purity of this vaccine used in that manufacturing process. Also, I'm showing you a size exclusion HPLC column. This shows that 99% of the vaccine is monomer. It doesn't have that tendency to aggregate upon itself, now forming dimers, trimers, et cetera, and catamers. Finally, most importantly, we've removed the N-glycosidase activity, and when we engineered out those residues from 199 to 267, this is a rabbit reticulocyte cell-free translation system where we can add messenger RNA for luciferase back into this cell-free translation assay. And what you see here is that when you use the recombinant RTA, this is the RTA that we haven't removed any residues from. This is the control. What you see is the nanomolar concentrations of the recombinant RTA. You get inhibition of protein synthesis. However, our vaccine, either with the loop that 3344 is still on, or with it taken off, we have no tendency to inhibit protein synthesis up to 4 micromolar of the vaccine. So what we've done now, we've engineered stability in the protein by removing those C-terminal residues, and at the same time, we've also removed the enzymatic activity of this protein. So we've addressed 2 of the issues that FDA had with previous candidates of this vaccine. This vaccine, when used in vaccinated non-human primates, again, our animal model is African green monkeys. When these monkeys are vaccinated with 80 micrograms of vaccine at 0, 4, and 8 weeks, and then challenged by aerosol 30 days after the vaccination, we see 7 out of 8 monkeys are protected from a large aerosol challenge to these monkeys. 3 months after, the group is completely protected. 6 months after, still completely protected. 12 months after, we have 3 out of 7 non-human primates protected from aerosol challenge. All controls die, and they die in very rapid time. And so we did a pivotal tox study in New Zealand white rabbits where these rabbits were given 4 doses of vaccine, 200 micrograms per dose, using the same schedule that we had planned in using in humans in the clinical trial. We saw no significant adverse events in this pivotal tox study when these rabbits were vaccinated with 200 micrograms, 4 doses. So we entered a phase 1 clinical study. This is the first time in man with this vaccine. The primary objective, of course, is to look at the safety of the vaccine. In this case, at 3 doses, 20, 50, and 100 microgram doses. The secondary objective is to evaluate the immunogenicity of the vaccine in escalating doses. So this is an open-label, uncontrolled, escalating multi-dose vaccine study. Group 1 and 2 received 3 doses 4 weeks apart. Group 3 received only a single dose of 100 micrograms of vaccine, and I'll explain that a little bit further as we go through the slides, why that was. 10 subjects per group. To ensure the safety of the subjects, there was a dose administration proceeded in staggered fashion for the first 3 subjects. So the demographics, 67% were male, 97% were Caucasian white. Again, 20 microgram dose, 10 subjects. All 10 received all 3 vaccinations at 0, 28, 56 days. Group 2 received 50 micrograms at, they received all 3 doses, again at the same schedule, 0, 28, 56 days. Group that received 100 micrograms of the vaccine received only one dose, one single dose. So related systemic adverse events from this study, most of these, and when I say related, that is related to the vaccine. There were a lot of other adverse events that occurred that were not related to the vaccine. 80% of the subjects reported at least one type of adverse event. We had, and most of those were mild, 67%, and of the 67%, most of them were headache, 30%. We had 4 severe adverse events. One of the subjects in the first low dose group had a severe adverse event, systemic adverse event. And I'm going to talk about this a little bit later on because this is also considered, the clinician also considered this a serious adverse event. Serious adverse event is reportable to the FDA, and I'm going to talk about the subject a little bit later on. 2 in the 50 microgram dose group had what the clinician reported as severe adverse event, and one was a headache, one was diarrhea. In the 100 microgram dose group, again, we had one subject that reported a severe adverse event, and that was a headache as well. We had 2 subjects that had grade 4 adverse events by I keep pressing the wrong button here, sorry. These grade 4 adverse events by Code of Federal Regulations in the clinical protocol are life-threatening, and I'm going to talk about these 2 cases as we go further. In terms of related local adverse events, we had 57% reported a local adverse event. The majority of these, 50%, were related to the injection site pain. We had, in terms of a severe local adverse event, we had one subject, and the same subject was the same one on the previous slide. I'm going to talk about her, I think, in the next slide. So these are the 2 grade 4 AEs. These are the life-threatening AEs that the clinician reported by FDA guidance, has to report. And so these 2 males had increased CPK levels. Now, they were completely asymptomatic, but when they showed up, so when they were vaccinated, they had bleeds on days 1, 5, 7, and so forth. The first male had an increase in the CPK level the day after his first vaccination at 100 micrograms of the vaccine dose. He was counseled and told to hydrate, and so he drank a lot of fluids, and this resolved the issue. And so the duration of the elevated CPK was 8 days, but this peaked at 3960. The normal upper limit for normal man for CPK is about 384 units per liter. This is well above it, and the triggering point is 6-fold above that. Then the clinician has to refer to this as a grade 4 AE. Now, the second male had a different situation. And before I tell you his situation, these are first cousins who worked in the same location and did the same job. They were first cousins on the maternal side. This individual had his first vaccination. His CPKs were all normal. He showed up at the clinic on day 28 for his second vaccination. He was bled that day, and his CPK level was 463. So the vaccination was held. They kept checking the CPK levels, and they kept rising. And ultimately, this CPK rose to 30,718 units per liter. Now, this was on a Friday, and because of the weekend, he wasn't able to hydrate himself enough to lower the CPK, so they admitted him into a military hospital. As soon as they admitted him to the hospital, this became a serious adverse event reportable to the FDA. And so he was hydrated in the hospital with 7 liters of normal saline, and his CPK levels came down, and he was released on Sunday. Interestingly enough, the onset was 28 days post-vaccination 1. And the night before he came in, he moved some very heavy furniture, which people who are getting vaccinated are told not to do. You don't do strenuous work before you come in for a bleed to do prior to vaccination, because strenuous work increases the level of CPKs. So the clinician is saying, in terms of whether something is related to an adverse event, and you have categories of that, likely, possible, probable, definite. And the clinician here said possible relationship to the vaccine. We have not proven that that was the case. There is a number of genetic disorders for CPK. The CPK levels here were the M dimers, so it was skeletal muscle that was being degraded. This person here did have myoglobin detected in the urine as well as the blood. So the two serious AEs reportable to the FDA was the person, the subject in the low-dose vaccine. This individual, a female, came in and had her vaccine in her right arm, and she had tremendous pain in her right arm after the first vaccine. And so she was given ibuprofen and sent home. When she came in for her second vaccine, it was given in her other arm, her left arm. She had no problems with that vaccine. On the third vaccine, she came in and she had vaccination back in the right arm. And then she had tremendous pain, arthralgia, she had pain under her armpit. What this individual didn't tell us was that she had two injuries to that shoulder. She had an injury skiing and she broke her shoulder, then she fell off a horse and had another injury in that same shoulder, and she never reported that during her history. And I think that was part of the problem. We would have never vaccinated her in that arm if we knew that information. So she had what's called SERVA, it's in the literature, shoulder injured related to vaccine administration. The relationship here was definite. We could have given her placebo in that arm and she would have had the same reaction she had with the vaccine. And this was the male with the rhabdomyolysis that I discussed earlier that had to go into the hospital because his CPKs were above 30,000. And so I saw this subject about a week ago. She had surgery in her shoulder, and she's doing fine now. This individual has normal CPK levels, and he's doing fine. But FDA did not want us to administer any more vaccine at a 100 microgram dose, and that's why you only saw the 10 subjects receiving one vaccination at a 100 microgram dose. And so in terms of immunogenicity, the ELISA geometric mean titer is the following. So we dilute the sera 1 to 500, and anything over a titer of 1 to 500 is considered positive. The grayish area here represents the vaccine dates of 0, 28, 56 days, and you can see after that this person, this group, both group that received the 20 microgram and the group that received the 50 microgram dose, you see a large increase in the ELISA titer after the third dose. With the toxin neutralization assay, the geometric mean corresponded pretty much as the ELISA titer, and we saw an ELISA titer, we saw a neutralization titer over 1 to 50 after the third vaccination, which slowly came down. We had 4 subjects that agreed to be boosted, and they were boosted 20 months after the first injection, and the response was, the immunostic response was significant. These titers just rose up and kept very high. They're still high now, except for a few that are coming down very slowly. This individual, number 2 here, right here, did not even have a TNA titer on the primary series. But this individual obviously has a titer now. And the other 3 subjects, again, their TNA titers were very elevated and remain elevated. These 4 subjects agreed to be in plasmapheresis at the NIH. We've been able to generate about 7 liters of their plasma. Now we're doing passive transfer studies with purified IgG from their plasma. And so in conclusion, this vaccine appears to be safe at 20 and 50 microgram doses, as well as being immunogenic. The single RVEc booster elicits a robust immunostic response. The booster appears to be necessary for producing high levels of neutralizing antibody titers, as most protein vaccines require. We've been funded to do a second clinical study, and this study is going to look at IM versus ID vaccination, as well as a different schedule where we vaccinate at 0, 28 days, and then boost. The third primary series is at 106 days with a boost at 12 months. And we'll also be looking at different doses. Instead of the 100 microgram dose, which we can't administer anymore, we're going to be looking at a 75 microgram dose, the 50, and a 10 microgram dose. I'd like to acknowledge the funding from DTRA, the Joint Science and Technology Office, for ChemBio Defense. And if there are any questions, be glad to answer any questions.